So, image analysis is another one of the very important aspect of the two-dimensional electrophoresis workflow. There are different type of image scanners available from different vendors, such as one I have shown here, molecular imager, densitometer, other the Typhoon variable mode imager. There are many staining image scanners available. So, now how to analyze these images? Do you want to do the things manually? So, can you take your gel patterns and sit two of you together and say, okay, this is my protein in control, this is your protein in treatment. Now, I am going to look at each spot manually and then going to size the spot based on this comparison. So, that is going to be very tedious work, and you will not have any information about the whether your spots having any statistical significance or not, how reproducible those are. So, you need to scan it by using good scanners, and then finally, you need to analyze your image from different softwares which are available. So, there are commercially many softwares which are available for doing two-dimensional gel analysis. I will give you a comparative table at the end, but almost all of this software, they take the scanned images and analyze your gels by using various steps. So, all these software enable the spot identification, comparison of the gels, overlaying of the images from your control and your treatment, cropping the gel, the part of which you want to compare, and further doing the statistical analysis. So, the crop tool, that is the first part which you would like to use. If in your gel you have some extra regions which where you do not have any of the spot of interest, probably you would like to crop those regions and crop both your control and treatment gels uniformly. So, this crop tool allows for a very specific defined region of the gel to be cut from the entire gel. It helps to select the region with the high spot density, which can be used for doing further gel analysis. Next, you would like to see your spots in more detail. So, you would like to use zoom tool, which can expand a specific area of the gel for doing further analysis. Now, image overlaying is very important aspect, because if you are comparing two gels, on one you have control, other you have treatment, you would like to overlay those images together to compare the spot pattern present on two different gels, because you have acquired two separate images. Now, you need to overlay those so that it can appear as a single merged image. Now, spots which are going to coincide on top of each other, whereas you can also locate their original position from the each of the individual images. So, image overlaying is important aspect, where you can merge your control and your treatment gels. Then you would like to do the spot analysis, where it is possible that you can obtain physical and statistical parameters for each spot which is present on the gel. You can look at the three-dimensional views of each of the spot, how they are differing from the control to the treatments, and then you allow the comparison of the gels spot by spot basis. So, often running a gel or acquiring images and generating lot of data is much straightforward as compared to doing the analysis, which is more tedious step. One has to really sit and go through the gels, usually spot by spot, to analyze the gels. Now, I am showing you a gel pattern of control and its comparison with the treatment gel. These are taken from one of the healthy control and a disease sample, and each of the spot is compared from the control to the treatments, and one can look at from different healthy controls and different disease sample what is happening to each of the spot, and if there is statistical significance for their overall change, if it is going up or going down, is that uniform in all the gels and how significant that is. So, all of this analysis can be performed by using different softwares. As you can see in this image, I am showing you one spot which you would like to compare across 16 gels. Now, you have zoomed into that particular region, and further you are looking at the 3-dimensional pattern of each of the spot, how they are differing from the control shown in the black and the blue spots, and the treatment which is shown in the on the black background in the red spot. So, after looking at 3-dimensional views of this particular protein, you can confidently say that this protein expression is changing and it is going a higher amount in the treatment. Now, one can look each of these spot intensity in much detail, and then followed by plot different type of parameters for percentage volume or spot intensity to compare their values and do the statistical comparison. There are various commercial softwares which are available for comparing the 2D gels, such as ImageMaster 2D Platinum from GE Life Sciences, PDQuest from Biorad, Delta 2D from Decodon, Dimension from Syngene, Ludici 2D gel image analysis software, Progenesis from Nonlinear. These are just a very few number of software which I have mentioned. These are which are very commonly used, but there are many other good software also available which one can use to analyze these gels. Now, let me show you this animation for performing 2-dimensional gel analysis, how to go step by step to analyze your gels. 2D gel analysis software. In this animation, I will describe you how to analyze the 2D gel images by using a generic software layout. So, first you need to load a gel image. It is possible to load either a single or multiple gel images simultaneously. This can be done by means of the load option in the file menu. You can save the gel images and then you can crop the area depending upon what area you want to analyze. There are several tools which are available for the analysis of gel. It is possible to crop the gels by selecting a specific region that is to be studied and then selecting the crop gel function. Cropping gel helps in selection of region with high spot density or to reduce the regions which contain high background stains with no spot. Zooming into a selected region. If you want to expand a specific region, you can use zoom tool. A specific selected region of the gel can be zoomed into for viewing the spot more closely and for comparison of spots between the two gels. This is particularly useful for gels with large number of spots. Once you have seen the area, you would like to overlay the images. Overlaying of images is a particularly useful tool for the comparison of two gels. The gels are overlaid such that they appear merged and spots that coincides will overlap with each other. This is extremely helpful while comparing the large clinical samples of controls and treatments, so that you can obtain the clear indication of the proteins which are differentially expressed. Now, After the analysis, one can look at the graphical representations of these three-dimensional view of the spots. The spots on the gels can be displayed as three-dimensional graph. Either the entire gel can be chosen or a particular region can be selected for this representation. The peaks obtained in the graphical representation are directly related to the spot intensity. Next, we will talk about spot analysis. Every spot on the gel can be detected by selecting the detect spot option. Various parameters such as smoothness, saliency and minimum area must be suitably adjusted for maximum clarity. Once this is done, each spot will either be encircled or marked with a cross, depends upon the setting along with the spot numbers. Now, I will describe you the gel matching. The software facilitates interpretation of the gel images by matching two different gel images which were obtained in your experiment. The matching spots are marked and after matching is done, any variation in the spot intensity, spot positions can be indicated by the blue lines as shown in the animation. This provides an understanding about the reproducibility across the gels. Once you have analyzed the gel, one can obtain the detailed information for these spots from a spot table. Information regarding various physical parameters of each spot can be obtained via this spot table, which provides spot number, intensity, area and volume of the spots as well as the saliency of spots. These parameters help to judge the quality of a gel. In addition to the physical parameters, various statistical parameters can also be computed for each gel and each spot on the gel, such as central tendency, mean, median, dispersion, coefficient of variation, standard deviation or other statistical parameters. The scatter plots and histograms can also be plotted for clear data analysis. These can provide information regarding inter and intra-gel variations. The spot comparison. It is possible to specifically compare a particular selected spot across the gels. When the gel is run with molecular weight markers with molecular weight of unknown proteins can be estimated from this information. For example, as you seen in the animation, on the left hand side you first loaded the molecular weight marker, and now from that information you can compute the information for the unknown protein to calculate its molecular weight and isoelectric point. These parameters, in addition to the other physical and statistical parameters, can be obtained for each spot. today I have invited a guest here to discuss the image processing and data analysis by using commercial software. So, today we will have Dr. Srinivas from GE Healthcare, who will discuss the how to analyze gel by using Image Platinum software. So, welcome Dr. Srinivas, and we would like to initiate discussion with you on image processing.
Yeah, this is the software layout where you can see where in your PPTs. This is the original software layout. Now here there is an option like image loader. Like add it. Wherever your gels stored, you can go to that place and add the gels. Like as I have multiple gels here, I am going to add few gels to our software to analyze. Like gel 1, gel 2, gel 3 and gel 1, gel 2, gel 3.
So, you are processing 6 gels.
6 gels at a time. These are all the 6 gels. Basically, 3 of 2 replications, like 3 replications for each gel actually.
Right.
That means one is control and another is the treated in that way. Now, as we discussed in our PPTs, this is the thing which is asking for particular staining. Here we can give chromacy or silver or different fluorescent. So as this stained with silver, I am giving this silver and I am applying the same color for remaining the gels. Now click on OK. Now as you can see the 6 gels at a time in this overview. After that, now you can see the proper spots in all images, very bright images of which you can see. So now we need to edit the images for visualization.
So, we need to change the contrast
and brightness, different features. So this is the button where you can adjust contrast and brightness. So as I can show here, if you now you can't see any differences now after changing also, but one can have to press on apply. Another feature is available here.
So now more spots are visible and
more spots
more
are, yes, more sharp. Otherwise there is an option that you can select a particular area, okay, then change the parameters. Now you can see real-time changes in that particular spots. Now display them. Now you have all of your gels here, okay. Once you have the gels, we can directly detect the spots now. Okay. From this button you can detect them. Now the 3 parameters: smoothness, saliency and minimum area. The smoothness, as we can see, if you are increasing the smoothness, it is going to under splitting, and if you are decreasing this smoothness, it is going to over split. So it is inversely proportional from the splitting. So it's a average value from 1 to 5 actually. So but one can set as a 2 to 3. It is sufficient. This value is sufficient for
to
start actually. And saliency, again this is also one can set up to 100, 150, but to start very good value is almost all 15 to 30. Okay. I am just giving 15 for this initially. Then minimum area 5 is quite good enough. If you think in your spot there are more spectacles, like there are much artifacts are there, you may increase this also. In that case you can be remove very easily more artifacts. So in this case, otherwise 5 is okay, fine value.
So all the 3 parameters now one can actually accept those.
Exactly.
And then detect the spot.
Exactly. That is what the software detecting the spots. Now it's detected all spots in this gel. If you are satisfied with this particular detection, then it's fine. Otherwise, one
maybe
can.
We should zoom in my region and see like.
Yes, exactly. I'm going to do the same. And now you can zoom this particular region. See how this spots are boundaried, as well as how the detection is there.
Can you apply the
whether-
same zoom in parameter for all the 3 gels?
Yes, definitely. First come back to original. Now again,
Let's select one region and then apply that for all the gels.
no need, you just select like the same region. It is going to got it.
Right. I think it's very useful to see this way because now
Exactly.
I think the parameters are very well defined
Exactly.
because it has properly defined the boundaries.
Properly define the boundaries.
And there are some spots where one can actually tweak around to get the
Yeah, exactly. If you want to edit these particular spots,
right.
whereas you can see there is, I think you can't see there is a protein, but it is showing as a protein. One can delete this kind of spots also.
Right.
Like one should have to go to edit the spot. There is an option like edit enable.
Right.
Go there and just delete the spot. Wherever the spot you would like to delete, this particular spot I am not interested in this, then I can delete it. Then,
But before we take the decision, can we just have a look on the 3D profile
yes,
of this
definitely.
one to ensure that we are not actually deleting a
Yes, definitely.
real spot, a faint
Definitely,
spot.
definitely. And then reports, go to 3D view.
So the spot which we are interested in discussing, that is highlighted with the green.
Yeah,
boundary.
green boundary, but it is only present in this particular spot, but it is not there. And but we can see some portion here actually, that particular spot. See, but that is why it is detected there also.
Right. So I
Yeah.
think sometimes it is possible like your
yeah.
Treatment may have that spot
Yeah, exactly.
appearing due
Yeah.
to the upregulation of a protein,
Yeah, exactly.
or it is totally shut down. Because
But
of that.
but it is not the case exactly here, because these are all the 3 replication of the
Okay, right.
particular same protein.
So this should be the same.
This should be the same,
Right.
but this somewhat it is not there, so we can delete these 2 spots here exactly.
Or in this case, I guess it is doubtful, right? So
Yeah, it is doubtful, or let it be. We can then finally we can see the statistical data whether it is there in both gels as well as treatment also,
Right.
so that one can go with the statistical parameters.
right.
This will be helpful there. So no need of deleting also in
Now
this
let's look
case.
at in detail about one of the real spots.
Otherwise, I can visualize this thing. I can more zoom so that one can see more. Yes,
So now this is the intense spot.
intense spots which we are visualizing. As
Another boundaries are marked from the 75% from the top.
Yes.
Now let's say we can actually zoom out and
Yeah.
go back to the whole gel. So let's say if in a region we have missed out some spot, like software has missed out,
Yes.
then there is an option to add the spot as well.
Exactly as well. This is the adding. Like let me zoom. Let me select a particular area. I'm interested in this particular area. I'm selected everywhere, and let me zoom that. Now you can see this particular area in all gels. Okay.
Right. I think it has defined the spot boundaries quite well overall.
But yes.
So that's why it's better to rely more on the automated
Yes, exactly. That is why.
automated detection.
Exactly. This is what. If you want to if you want to include some spot wherever, if you are interested, you can definitely add. Like this kind of extra portion, we can as easily we can delete actually. Like we can reduce this particular portion.
Second better ways to remove the spot and then draw the spot.
Yeah, that is also better way. But
Yes.
both ways you can do actually. Yes, you can delete it or you can redraw that. Is both as well as we can do. Now we find all spot boundaries are okay and everything is fine. Now we can match these 3 gels. How our reproducibility of our 3 gels, how reproducible of our 3 gels. Okay, this is what which we can do here. So come to your original state without zooming and match them. This is the match option.
Okay.
You can match them.
So now you are applying the same analysis parameter on both control and treatment
Yes,
gels, and you are
exactly.
matching
Yes,
the gels.
exactly. Now we are matching only control gels only.
Only one group.
Only one group. How our reproducibility is there within
one group.
within one group. This is what we have to check initially.
Okay.
So if you have, suppose maybe instead of 3 replications, if you have maybe 5 replications, maybe 6 replications, out of 5-6 replications, one can remove that particular gel and they can take remaining 5 gels.
So if there is some time during various experimental run, if there is 1 or 2 gels which are very bad, which are going to affect the overall reproducibility.
Exactly.
So by looking at this type of parameters, one can decide, okay, out of 6 gels, maybe 1 of that is very bad and going to affect our statistical parameter.
Exactly.
So maybe one should remove that.
Exactly.
And then select those ones which are having the very good number of matching.
Yes, that should be done. And now we can see in these 2 gels there are few vectors which we can see. This we can able to remove by adding the landmark sort of thing. Okay, now this is the landmark option. Okay, you have to add your landmark initially in the reference gel only. Like if suppose you can think this is a landmark sort of thing, I'm just going to add a landmark here. So I have to adjust this landmark to the same position in remaining all gels.
Now vectors are, I think, improving
yes,
based on the landmark position.
Now again match them.
So from 146 it has increased to 746.
Exactly.
By increasing the
by adding
a landmark.
A landmark.
And now vectors are matching well.
Yes. See.
Right.
But you can see there are few
vectors,
but still there. But these are all moving at the same side. So that means there is not like irregular.
Also, I think one need to go through each region
very carefully. So if you can add some more, one more landmark here, it can be removed very easily. Right. This is what we can do. Okay, now we can save this particular thing and apply the same parameters to our treatment gel also.
I think that's important because you don't want to make any changes between your controls and
treatments. So you can see no need to remember also the same parameters. If you can detect your spots, it automatically gives the same parameter earlier which you have used. Okay, so same smoothness is 3 and saliency 15, minimal area is 5. So no need to change anything. Then click on OK.
So is there a long process of analyzing it, or another? I think sort of quite quick.
It is quite quick. Then it's completely finished. Now the same way you can go through each and individual gen by zooming and 3D view, and you can select which are all the spots and which are all the not spots. Then you can directly match them. Now I'm doing the same matching. 1274 matches have been done. So these gels are more reproducible. That means as compared to our control. Yeah. See, there is no even vectors also. Without giving landmark also there is no vector. Right. So this is very fine gel. So now what is the replication which we did was confirmed. Okay, these replications are okay. Now one can analyze in the class analysis in between these two classes, like interclasses and intra-class sort of thing.
So between a control and treatment group, one can actually now do the statistical analysis.
So now I am creating the classes here. Otherwise, before that I would like to go to one report point. What is the gel table? Okay, that is the these are all the gels which are present here. Shift A, you can select all the spots. Go to spot table. See now.
So what parameters are displayed here in the spot table?
This is the file name and the spot ID. It has given a particular spot ID for all proteins, and this is the pixels. It is the this is the match ID. That means out of 6 gels, this match ID which is giving. One second.
Just continue quickly. Like what are the?
This is the file name. This is the gel number. Then spot ID, which is given complete number for all spots. Then match ID. then this is the pixel size and x-axis and pixel at y-axis. Then if you can get able to give the PA also, and you can calibrate the PA as well as molecular weight also, then intensity of particular spot and area of that particular spot and volume of that particular spot and percentage volume of that particular spot and saliency of that spot. These are all the things which you can see in your
parameters for all the gels can be obtained from the
software. These are all the parameters which you can see in the software. Okay, so if you are satisfied with all these values, then you can go ahead your next level of analysis, that is class analysis. Okay, now I'm going to create a class here. Create a class. This is the 81. Another is the 82, which is control and treated. Now again you can shift these gels into 81 to 81. Another match set which I am creating, so where we can easily move easily before going to class, so that
you are moving all the 6 gels
into composite match set. So all 6 gels are here, and now you can create again the same way classes.
So by adding different folders, I think one can actually avoid any sort of carry or mistakes in previous one.
Yes, exactly.
And
so that
all the steps are stored.
Yes, all the steps are stored. Now again I am shifting all 82 gels into 82 folder. This is the class folder again. 81 folder. Now
this is for the class analysis.
This is for class analysis. Now we can see both gels together. Now 3 gels from the control and remaining 3 gels from the treated. Okay,
right.
This is what which you can see. Now it is taking values as a single gel of 81 and single gel of the 82. So now it is going to average of these 3 gels, and now it is another averaging these remaining 3 gels. So a single value you can get from the control, another single value you can get from the treated. Once you did the same thing, then quickly match them again. One can remove all these vectors by adding our particular landmark. So
this is
the same process what we did earlier. Now go to reports, where you can see the analysis classes. Go to table. Now you have the match ID. The match ID is the particular spot number as well as the maximum area. This is a complete maximum. Whatever the maximum area, as well as this is the 81 value and 82 value. Okay,
so for the control and treatment,
yeah,
both groups we had 6 gels, right?
Yes.
So what are the values in each of the 6 gels for each spot?
For each spot, exactly. This is the averaged value again.
Average of all the 3.
All the 3 gels
from each group.
Yeah. Ratio value, if you can see, this is the ratio value which is giving the upregulation or downregulation. Okay, and I think we need to add. Yeah. Now this is the statistical parameter on
the
basis
of software gives you option for doing different type of statistical analysis.
Exactly. So this is the ANOVA value which we can see. On the basis of this ANOVA value, we can easily select our interest of protein, as well as you have the fold regulation also. This is 1 fold upregulated and this is the 1.2 fold downregulated. The same way you can see the whole values. Now no need to go each and individual spot. You can filter them easily. Now what we are going to do is, first at least ANOVA value which we are going to take, the maximum we are going to take is maybe 0.05 as the statistical significant value. So those many spots it can be highlighted. Okay,
right.
So this is the spot which is already undergoing this particular
ANOVA value
threshold, and it can see all the spots which you have this particular this particular NO value, those are all highlighting here. Now it can be very easy, one can easily go through that, and this particular table you can easily export to Excel also. From there you can have your complete data also. This is what the complete analysis software gives you the output.
So I guess it was very useful to see like different steps what are required for performing this analysis, and also the software gives you a lot of options for doing different fold change and statistical analysis of how significant data is, and then one can actually still go back to those spots which software says is significant and look at manually each spot to verify that is a real spot.
Yeah.
So thank you very much, Srinivas, for very
Welcome.
useful discussion and demonstration on the software for image processing on two-dimensional gels. And we'll continue our lecture flow for further two-dimensional difference in the electrophorosis in the next class.